Welcome to the series of microbiology on one page. So, in this series, we are going to cover all the topics of microbiology system wise. How are we going to cover them? System wise. Okay, and we are going to write the entire chapter. Like, for example, today we are doing food poisoning, and we'll be covering that on one page. So, let's start with it, and I'll be sharing the notes later. Let's start with this topic called as food poisoning without any further ado. Okay, so what do we need to know in food poisoning? Firstly, food poisoning with onset. What is food poisoning? We will define it. Food poisoning refers to an illness acquired through consumption of food or drink contaminated either with microorganism or their toxin. So, it is an illness acquired by consumption of food or drink contaminated with microorganism or their toxin. This is a basic definition. Any food or drink which is contaminated by a microorganism or their toxin which is present in that food. Okay, this usually commonly results in two things: diarrhea, vomiting, and other GI symptoms. We'll study more about it. But when we study food poisoning, we classify it very broadly. Okay, so how do we classify? Classification of food poisoning. How will we classify? We'll classify it broadly according to its onset. According to its onset. The first one, where the onset is from, okay, so the onset is 1 hour to 6 hours of incubation period. That is from the ingestion of food to the arrival of first symptom. We call that as incubation period. So the first category, classification of food poisoning according to the incubation period, the first one is 1 hour to 6 hours. Then we have 8 hours to 16 hours. Later we have the organisms which take more than 16 hours. Here we are going to list various organisms and the disease that they cause. Generally, the question that comes in your exam will ask you to explain food poisoning, to classify it according to its incubation period and the microbial agents causing it, or it will simply ask you what are the microbial agents, but you still have to write it according to its classification. And then it will ask you to explain any one of the important organisms that I'll be discussing further. In this chapter, the things that we are going to discuss is staphylococcal food poisoning, bacillus cereus food poisoning, and later when we are done with the food poisoning, we are also going to learn about botulism, okay, in brief. So let us start without any further. 1 to 6 hours. The first one that we have is Staph aureus. Now Staph aureus causes symptoms like vomiting, okay, significantly vomiting, followed by diarrhea and abdominal cramps. The common food which it is associated with is ham, poultry, salad, mayonnaise, pastries, dairy products. We'll be studying about staphylococcal food poisoning in detail later on. The second one that we have is bacillus cereus. Okay, B cereus. Next you have is 8 to 16 hours, having incubation period 8 to 16 hours. The first organism here is Clostridium perfringens. The second that you have is B cereus. Now you will wonder why I have written B cereus twice. So B cereus, it has two types. B cereus food poisoning has two toxin. Okay, its action is toxin mediated, and it has two toxins. One is diarrheal and the other one is emetic toxin. The diarrheal toxin is the one that is causing food poisoning. Okay, and that has incubation period 1 to 6 hours. So diarrheal toxin will have incubation period of 1 to 6 hours, whereas B series emetic toxin will have incubation period of 8 to 6 hours. Okay, you will further understand that. Then the organisms that are taking more than 16 hours, majority of the organisms take more than 16 hours. Under that comes first our very famous Vibrio cholerae. Then we have enterotoxigenic E. coli. In the upcoming chapters, in the upcoming videos, we'll also be discussing about enterotoxigenic E. coli or enterohemorrhagic E. coli. Basically, we'll be talking about E. coli. It's a long list. Okay, so this is the complete table. I've tried to list most of them. Okay, not all, but I've tried to list most of them. So first we have Vibrio cholerae, then enterotoxigenic E. coli, enterohemorrhagic E. coli, non-typhoidal salmonella, Shigella. Then we have we then have Parahemolyticus, C. jejuni, Listeria monocytogenes, and norovirus and myxotoxicosis. All of them take more than 16 hours. Now you don't need to list all of them in your exam, but you need to mention most of them. Okay, now let us talk about Staphylococcus aureus in detail. As I said, let us discuss that. Okay, so S. aureus, okay, is the name of the organism. So staphylococcal poisoning is usually, it is caused by contaminated food, okay, from infected human carrier. Okay, obviously it is carried by food and water, but what kind of food and water? Like what kind of food? We will talk about that. Okay, so most common food item, food. But what kind of food? What kind of food? It can be spread because of ham, poultry. We have salad, mayonnaise. We have pastries and dairy product. Okay, so that is it. Then next is enterotoxin. How is its action mediated? Its action is mediated with the help of something called as enterotoxin. So if food is left for a long time after cooking, the organism gets opportunity to multiply. This food originally will contain S. aureus, Staph aureus. Now what will happen that whenever the cooked food is left for a longer time, this organism will multiply and produce toxin. Organism will multiply and produce toxin in food. What will happen next? The action of enterotoxin will take place. Enterotoxin is expressed by nearly 50% of S. aureus strains. It is preformed toxin. This is very important to know about enterotoxin. Enterotoxin is a toxin that is any toxin acting on your enterohepatic system. Okay, that in your GI system. Enterotoxin is firstly a preformed toxin. Very important. It is already formed. So nearly 50% of S. aureus strains uh, express this enterotoxin. In majority of them, in all of them mostly, it is preformed toxin. It is already formed, secreted in food, and then, okay, before consumption. It doesn't get secreted inside our body. Before we eat the food, it is already present in the food, okay. So it is secreted before the consumption of the food, so that it can act rapidly. Therefore, it has rapid action. We have seen that its incubation period is literally 1 to 6 hours. Rapid action. Therefore, incubation, we'll write it in the next point. Incubation period is 1 to 6 hours. That quickly and rapidly it can act. Next up, we have site of action. Where does it act? Why is vomiting the most common symptom? Because this toxin will stimulate, okay? Toxin will go and stimulate vagus nerve. This vagus nerve, okay, plus the vomiting center, plus the vomiting center of the brain. So therefore, it also causes vomiting along with diarrhea. How does it cause diarrhea though? It will stimulate the intestinal peristaltic activity. Now once that is done, symptoms. It will produce some symptoms. We already know vomiting and diarrhea are the major symptoms. Along with that, what else can be the symptoms? Okay, so symptoms are the clinical features with which a person presents to you. A person will have nausea, vomiting. We all must have once experienced this in our lifetimes, okay? Nausea हो रहा है, vomiting हो रहा है, all because of consuming some old food, okay, undercooked food, some probably all the mayonnaise or ham, poultry, pastries, dairy products, anything can lead to this. So nausea, vomiting, all. Occasional diarrhea, okay, occasional, not every time, but occasionally it can also lead to diarrhea. It can also cause hypotension. The hypotension is purely because of dehydration, okay. So we also need to mention dehydration. The most common source of the food is food handlers, okay. They are the carriers of this. So carriers are food handlers, okay. The people who are handling the food, they are the most common carriers, and there is no secondary spread, okay. So for once a person gets it, it cannot spread again. Now we have to talk a bit about the serotypes, okay, about the serotypes of Staphylococcus aureus. What are the serotypes? For detection of this, whenever we do lab diagnosis, generally lab diagnosis is not done in case of Staphylococcus. It's a very, very common infection, but we still need to know the serotyping. So when we talk about serotyping, there are some important things. That first of all, there are 15 serotypes, and type A is most common and it causes food poisoning. Type A is most common and causes food poisoning, okay. Serotype F does not cause food poisoning, but it causes toxic shock syndrome. Type F, it causes toxic shock syndrome. And here the detection, okay, how do we detect? Detection of enterotoxin in the food is carried out by ELISA and latex agglutination. So ELISA and agglutination. So this is all about S. aureus. Okay, we will then talk about Bacillus cereus. As I said, let us talk about Bacillus cereus. Very interesting thing, Bacillus cereus. Bacillus cereus infection, it is normally habitant in the soil, but also widely isolated from food items. What food items? Vegetables, milk, okay, cereals, spices, meat, poultry, anything. But Bacillus cereus, very important source of like Bacillus cereus, B cereus, is fried rice. Okay, it's rice. It is caused by consumption, especially the emetic toxin. It is due to consumption of contaminated fried rice. Okay, so let us talk more about it. The one that is causing B cereus. Okay, the one that is causing this food poisoning, B cereus, it is a gram positive spore bearing motile microorganism. What is it? It is gram positive spore bearing and motile microorganism. Its action is mediated by producing two type of toxins. We saw that Staphylococcus is producing one type of toxin, but B cereus, it is producing two toxins. The first one is the emetic toxin, and the another one is diarrheal toxin. So let us talk firstly about the emetic toxin. Okay, then we will talk about diarrheal toxin. Emetic toxin, it is heat stable preformed toxin. Therefore, fried rice, it is present in the fried rice. Rice is cooked, but it is not killed by heat. So it is heat stable toxin, not killed by heat. Okay, so how much ever you cook the food, it won't be killed, it won't be destroyed, the toxin won't be destroyed. It acts immediately on the intestine, so its incubation period is 1 to 6 hours. Okay, incubation period is very short. Why? Because the toxin is already formed. It's a preformed toxin. Heat stable, I'll mention it over here probably. Preformed toxin. Later, it is due to very important consumption of contaminated fried rice with emetic toxin. The heat resistant spores of B cereus, so it is not resistant to heat. Okay, it is resistant to heat. B cereus, my bad. B cereus spores are resistant to heat. It is not destroyed by heat, and they can survive boiling. Therefore, they are present in the rice. As I said, if cooked rice is not refrigerated, the spores can germinate and produce toxin. So what happens is, whenever we cook the rice, they are not destroyed. And if the rice is not properly stored and refrigerated, these spores, these microorganisms, as we have already studied in Staphylococcus, the similar way they will also multiply and produce toxin in the food before consumption. Now when a person consumes this food, that person will get this severe vomiting. Okay, so frying before serving may not destroy the preformed toxin, and therefore the symptoms are produced. What are the symptoms? Same: vomiting, nausea, hypotension, dehydration, very common. There is second toxin that we talked about just now, that is diarrheal toxin, this toxin. So we will write about it. Diarrheal toxin, it secretes diarrheal toxin, organism secretes this toxin only after entering into the intestine. So therefore it acts late. Now you know that this toxin is released only after entering the intestine. Now you know why the incubation period in this case is 8 to 16 hours. Why it is 8 to 16 hours. The other manifestation of B series can include other manifestations. Other manifestations are very rare, but they can happen, but can occur. Which is the first manifestation? The first one is ocular disease. What ocular disease? Like severe keratitis. The next is vision loss. These are the ocular diseases. Second, it can also cause systemic infection. It can lead to meningitis. It can lead to endocarditis. It can also lead to osteomyelitis. 1, 2, 3. These. So this is all about Bacillus cereus. We have known enough. Let us talk about next, food poisoning with onset of 8 to 16 hours. We have talked about the organism causing this. One of the important organism between 8 to 16 hours is Clostridium perfringens. Be serious, okay? Clostridium perfringens, all you need to know is it majorly affects the skin, but it can also be it can be a causative agent for food poisoning as well. So Clostridium perfringens, it is spread because of inadequate cooked beef, meat, poultry and legumes. After ingestion, so what is it? Beef. We're talking about Clostridium perfringens. Inappropriately cooked or undercooked beef, meat, poultry, all of this, they can lead to Clostridium perfringens spore to multiply, because even that spore is heat resistant. All of this will contain the spores of Clostridium perfringens. They are heat resistant, so how much ever you heat it, they won't be destroyed. When it is not refrigerated properly, they will multiply, and after ingesting, the toxin will be produced. So here the only difference is that it is almost the same, just toxin is produced after entering body. This is the only difference. And the illness usually begins suddenly. It is self-limiting and lasts for almost 24 hours. So that is it. The other organism that are causing more, their action is taking more than 16 hours. Again the same, they will produce their action after entering the body. Okay, some will produce toxins, some will somehow multiply in the body and then affect various organs, Vibrio cholerae or E. coli, Salmonella. Everything will be studying more in detail in other chapters. So we are not focusing even a little bit on more than 16 hours. We directly come to the lab diagnosis of food poisoning. Okay, we talk about the lab diagnosis of food poisoning. So let us talk about lab diagnosis over here. Laboratory diagnosis. So meticulously history taking, okay, regarding the ingestion of any food, okay, before the onset of symptoms like diarrhea or vomiting, it is very important. Whenever a person comes to a physician, we generally ask what kind of food have you consumed, so that you are getting history of vomiting or diarrhea. It directly says that a person has some history of ingesting contaminated food. So we have to first of all processing of the food specimen. Now before that, viable plate count. Okay, what is that? It is standard method for direct quantitative culture. So food material, okay, it is homogenized in sterile diluent. So whatever the food material the person has consumed, we collect that. It is diluted in the Ringer's solution and then plated and in appropriate medium. That is one method. But what if the person says that मैंने तो खा लिया सारा खाना, I don't have any food left, okay, the food that I consumed, I don't have any of it, so the person does not have that specimen. So what else can be done? We can take other specimens, okay. For example, the stool specimen or the vomitus specimen, they can be enough. So we have 3 specimens over here. Specimen, what specimen? We can take either vomitus, we can take stool sample, or we will ask the person, okay, the suspected food that he has eaten, we can ask the person to get that food, okay, and then it is put in the Ringer's solution and then plated onto appropriate medium. Pre-enrichment cultures, so pathogenic bacteria from the suspected food, it can also be isolated on agar culture, okay, so that is called pre-enrichment. And what does that do? It helps in recovery of the damaged organism. So suppose the organism now is like there in the food, it is dying, it has no more, obviously it's a spore forming organism, okay? So the organism is now no more multiplying, it's dying, but when we put it on the agar medium, it multiplies and it again gives rise to spores. These spores will form the toxin, and therefore we will be able to understand which type of organism it is. So what we can do, first is specimen collection, then what we can do is viable, so sorry, something is wrong with the pen, okay, viable plate count, okay. For this we will add the suspected food to the Ringer's solution. And third, what we can do is pre-enrichment culture, okay. Pre-enrichment culture, basically what we do is we take the food, isolate food, okay, and it can be isolated on the agar culture. After pre-enrichment, okay, some enrichment, okay, selective enrichment is done to it, so it will revive, okay, the damaged organism, the already dying organism, it is revived because of pre-enrichment. Next is toxin detection. I have already told you how to detect toxin. First we do ELISA, or we can do latex agglutination. That is it. So that is it for the lab diagnosis. Now let us talk about botulism, okay. Botulism, it is caused by the agent C. botulinum. Now C. botulinum is a bacilli. B for botulism, B for bacilli. So C. botulinum is a gram-positive spore-forming obligate anaerobe bacilli, okay, obligate anaerobe. So therefore it is not generally, so those spores, okay, that are there, they need to germinate, and they won't germinate if there is aerobic condition. It needs an aerobic condition. Therefore in adult intestine it is rarely seen, but infant intestine it is very commonly seen, because the germination of the spores can happen in infant's intestine, okay. Now toxin produced here is botulinum toxin. Location, where is it found? It is present in the soil, it is present in the vegetables, it is also present in the sea mud. Pathogenesis, it has 8 serotypes. So what are the A, B, C1, C2, D, E, F, G? 8 serotypes. Now out of these, A is the most severe one. It is the most severe one. Whereas C2 is the only toxin, is the only serotype that produces enterotoxin. Rest all will produce neurotoxin. Let us talk about the pathogenesis now. Okay, since we know the serotypes, so we will talk about the pathogenesis. Entry of the organism. There are various mode of entry of organism based on the their like origin. We have divided it into various types of botulism. So let us first talk about the types of botulism. Okay, so pathogenesis start करने से पहले we will first talk about the types of botulism according to the place from where it is born. Okay, so first of all we have is types of botulism. First of all we have is foodborne botulism. Foodborne botulism is due to consumption of food which is contaminated with preformed botulinum toxin. Again, it is a there is preformed botulinum toxin in any food. As we know, the spores cannot survive in humans, in adult human. So there is preformed toxin, already formed toxin which we consume. So where is it found? It is found in canned, improperly, very important, improperly canned food. Improperly canned food will generally food and beverages, both of them will most okay, very commonly it will contain botulinum, C botulinum, okay, or Clostridium botulinum. Second, it is found in second it is wound borne. Okay, second type, it is wound borne botulism. Now wound botulism, what is it? It is like food borne botulism except for the absence of gastrointestinal feature. So it won't have the gastrointestinal features like constipation or any kind of GI effects like diarrhea or vomiting or any other features. It is simply having all the other features of flaccid paralysis, wound borne, and it is present in the in wound borne, the organism, it is present in the soil. It is present in the soil. From there it will enter the body. Next we have is infant botulism. Infant botulism, it is generally associated with consumption of food in infants, generally honey, in children less than 1 year. Therefore we say that children less than 1 year, they shouldn't be given honey. Okay, and the manifestations are like floppy child syndrome. Floppy child syndrome is characterized by various other symptoms like inability to suck. Okay, inability to suck the or swallow, weakness in voice. Okay, voice is very weak, voice weakness, and there is ptosis and floppy child syndrome and floppy neck. So the floppy child, the name comes from the infant's floppy, sorry, infant's floppy neck. From there the name comes floppy child syndrome. Okay, now let us talk about the pathogenesis. First, there will be entry of organism via the blood. Okay, it will spread to the peripheral cholinergic nerve terminals, peripheral nerves. Okay, and it will okay, that to the nerve terminal, peripheral cholinergic nerve terminal, cholinergic nerve. So its work is to produce acetylcholine, but this the toxin, it will inhibit acetylcholine receptors on the motor nerve terminal, and therefore it will cause flaccid paralysis. So just a few steps, okay, to learn. First, the entry of the organism via the blood, and that enters into peripheral cholinergic nerve endings that will inhibit the acetylcholine receptors present on the motor nerve, and that will cause flaccid paralysis. Now it is therapeutically used in spasmodic condition. Okay, strabismus and blepharospasm. All of these conditions we can use and this toxin, okay, this neurotoxin of botulinum as a therapeutic okay, mode of recovering from these conditions. But the recovery normally takes, if a person is infected with this, a normal healthy individual is infected with this, the recovery will take 2 to 3 months, because after 2 to 3 months new axon terminals sprout. The clinical features here, the incubation period is 18 to 36 hours. 18 to 36 hours. There will be clinical features like diplopia, dysphagia. There will be flaccid paralysis, decreased tendon reflex, constipation, respiratory muscle paralysis, which is very severe and can lead to death. So diplopia, dysphagia, there will be flaccid paralysis, dysphagia, there will be flaccid paralysis, decrease in muscle tendon reflex, constipation and respiratory muscle depression. Out of which flaccid paralysis is the most important one. So with this we have completed it. This is the food poisoning in one page. Okay, this is everything about it. Okay, thank you.